My name's Mark, and I'm from the National Heart and Lung Institute. You probably kind of gathered that from all the heart and lung stuff that you've seen today. And my job is I'm a pharmacologist. Does anybody know what a pharmacologist is, or what pharmacology is? Yes, very, very good, because it's derived from that. So pharmakon is Greek for poison in ancient Greek, or drug, okay? And logica is the study of, okay? So, or the knowledge of, actually. So basically what I do is to look at drugs and their effects on people, okay? And one of the ways, does everybody know what a gene is? Do you know what a gene is? No? Do you know what a gene is? You know what a gene is? Not the ones you're wearing. But okay, so yeah, genes are what makes you, you, as an individual. So have you heard of chromosomes? Okay, so in each of your cells, they have these chromosomes. And these chromosomes actually make you, you. So these are little bits of what they would call DNA, and they make up into these little sort of funny looking things, and there's 21 of them, and they're called chromosomes, and they decide whether we're male, whether we're female, whether we have blue eyes, whether we have blonde hair or black hair or green eyes. So they're the genetic material that makes you the person you are, okay? So as you can see, it's quite important, isn't it? Okay, and now are we all the same, or are we all different? Even different, even if you're brothers and sisters, you're completely different, aren't you? Yeah, okay. And what makes us different is our genes and how our genes react to the environment. Okay, so this is a study that we call, what we call bioinformatics. Oh, I'll show it. So it's a big word. Okay, so bioinformatics is the study of your genes, okay? And particularly we call this, I suppose. So over here, as you can see, nice sweets. Do you like one while you're sitting there? Okay, here we go. Is that sweet? There you go. There you go. Careful, these are my genes, so you might grow two heads. Okay. So would you like that one? Yeah. Okay. So the thing that makes us different. So you two will have similar genes, right? You three will have similar genes, but you're not all the same, are you? You don't look exactly the same. So what makes us different is how our genes react to an environment, to stimuli, and as a pharmacologist, what I find this is amazing to use bioinformatics is if you take a drug or if you have a particular disease, I can work out from looking at your genes and how they're working what would be the best way to treat you. So the reason we're doing this is it's heading towards a state called personalised medicine. Do you know what personalised medicine is? What do you think personalised medicine means? How friendly your doctor is? Go on. Brilliant, that's brilliant, yeah. Very, very good. I'm really impressed with that, okay. So, if I take some of these, so your genes don't always do nothing, right? Okay. So if I take, so we'll call these genes that go up, right? We'll add a few genes that go up. And we'll add some genes that go down, okay? Now, I'm not sure this is going to work properly now because if I told it too much. If I give this a bit of a mix around, I should have got a bigger bottle, shouldn't I? I'm just going to make lots of noise now. This is going to be great for your recording. Okay. So, now, does everybody, do you know, the thing about it is, not everybody reacts the same way to a drug, okay? And that's because of our genes and how they react. So the environment at this time is maybe aspirin or another drug that you're taking, okay? So for some people, it will work. For some people, it won't do anything. And for some people, I like to make them feel worse. You know, some people, if they take aspirin, hurts their stomach, right? Okay, so that's a bit like this. So what we're doing before, we used to just have a look at one thing. So we'd have a look at one thing, and that would go to that, and we'd correct it, and it would go to that. So it would go down. So we knew that this would happen. And there's lots of drugs that have been developed like this. So has everybody heard of propranolol? No. So propranolol is a drug that stops adrenaline beta blockers. So if you have a look in the panel book over there. So propranolol was the first beta blocker. It was discovered by a man called James Black. So James Black, and he was a really famous pharmacologist. He also, has anybody heard of cimetidine? Okay, cimetidine is for stomach ulcers. So it's a proton pump inhibitor. Okay, and that was designed that way. So in the future, we think we can design drugs for people, or put people on a combination of drugs, because there's you, and we can see a little bit, and then there's maybe your brother. He's different. And maybe there's your mum. Okay? So what we do now is we can then monitor all our genes. However, here's where the difficulty comes in. So if I come around here just a little bit, sorry. Okay, so if I put a number of these out. Okay, so you can see some genes have gone down, some genes have gone up, right? Okay. Now, say I correct these 3 genes, okay? That's good, isn't it? But that's not the full story. By correcting those 3 genes, I might alter these some more genes. So you see what I mean? So it's not that simple. So what we're trying to do now is we have a look at the genes. We look to see which genes are connected. So those genes and those genes might all be regulated or changed by this gene or this gene. And we correct those genes, then we get rid of all these. So that's the problem we have at the moment, is we're trying to find out what are the main genes, or what are the main genes that we need to change to stop a disease. So if I get rid of that gene, then we get that. So that's what bioinformatics is. Was that clear to everybody? Do you understand it? Okay. Thank you.